Hello, everyone. Good morning or afternoon, wherever you are. This is Christy Sullivan, and I'm the Secretary of the American Society for Cellular and Computational Toxicology. This webinar, like all of our monthly series of webinars, are presented by ASCCT and the European Society for Toxicology in vitro. And if you're a member of either of these organizations, you know that we have an interest in pursuing activities that promote and engage people in the computational and in vitro toxicology fields. We offer things like this webinar series, also annual meetings, trainings, and other activities. And both of our organizations try to support early career scientists as well. This webinar series, for those of you who are new to us, was bi-monthly for several years, or sort of bi-monthly. And you can see on our webinar archive on our website all of our past webinars. Now we've been able to transition to monthly webinars, and that's thanks to dedicated attendees like you and many presenters like today's presenter, Dr. Baker, who have been kind enough to donate their time and present their work. So lots of great things going on. Just some brief housekeeping. This webinar is recorded, and the recording will be posted on the ASCCT website, probably a few days from now. You'll also receive a PDF copy of the presenter's slides. Your lines are all muted. If you'd like to ask a question, I'm going to ask that you type your question into the question module on the GoToMeeting control panel. And during the Q&A after the presentation, we'll be able to ask at least some or most of the questions that are submitted. You can also reach us in the chat box if you're having some technical difficulties or have other questions like that. I just briefly wanted to mention a couple of things. First of all, the ASCCT is having its annual meeting. It's going to be in the fall, October 20th through the 22nd. And also, we have some great confirmed speakers. We just finished our call for abstracts and review, and we have really a lot of amazing science that's going to be presented. So I would encourage you to attend. It's virtual, of course. And as well, we have training on the EPA chemical dashboard tools on the Friday of that week, October 23rd. We're going to open registration on September 1st, so look out for that on the website. We also have some upcoming webinars in September. We have Joanna Niffler, which is very exciting. She's going to be talking about phenotypic profiling for high throughput chemical screening. And October, we're still getting scheduled, but it may be on fish acute toxicity methods. And as I said, you can view you can register for the September webinar, and you can view the archive on our website. Also, I wanted to mention that the European Society for Toxicology in Vitro has a congress. Their 2020 conference is rescheduled to 2022 in Barcelona in June, but most importantly, they have a training in October. And if you go to their website, you should be able to find links over to that training. So finally, to get to today's presentation, Dr. Nancy Baker is going to be talking about PubMed Abstract Sifter, which is a literature informatics tool. Dr. Baker is a literature informatics researcher specializing in finding ways to make scientific literature tasks more effective. She has a PhD from the University of North Carolina at Chapel Hill through the School of Information and Library Science and the Program for Bioinformatics and Computational Biology. As an employee of Leidos, she has worked as a contractor at the US EPA Center for Computational Toxicology and Exposure for nearly 10 years. Just one brief note before we turn the floor over. For those of you who would like to at the end and have time to stay after the presentation and the Q&A, Dr. Baker has been kind enough to offer sort of a hands-on tour of the Abstract Sifter tool. And I sent an email reminder this morning with instructions on how to download the Abstract Sifter file. If you didn't get that email or can't find it, you can also go to the GoToMeeting control panel. On the bottom there, you will see a handout. And if you open that PDF handout, while the presentation is going on, there will be instructions in there to download the tool so that you can be ready to follow along if you like. Okay, so without further ado, I'm going to go ahead and give Dr. Baker the floor so she can give her presentation.
Okay, great. Okay, Christy, can you hear me okay?
I can hear you, yes.
Okay, and here's my screen.
Yep, we can see your slides.
Okay, great. Thank you so much.
You're welcome.
Good morning, everyone. I'm Nancy Baker, and I'm here to talk to you about a tool I developed called the PubMed Abstract Sifter. My goal today is to introduce this tool, and the format of the way I'm going to do that is I'm going to give you a short PowerPoint introduction, and then I'm going to actually demo the tool. And then, as Christy said, I will stay on the phone as long as anybody wants to, and you can download the tool yourself and fire it up and practice if you wish, and ask me questions. I've never done anything quite like this before with this many people, but we'll see how it goes. And this is the download link. It's also in that PDF file that Christy sent out to everybody. Before I get started, I want to make sure I thank some people. Tom Knudsen is my mentor at the EPA. Two other programmers who have helped with the code in the Abstract Sifter are Tom Transu and Audrey Wilkinson. And of course, thanks to Christy and Bethany for inviting me today and helping get this all set up and running smoothly. I'd also like to thank all of the Abstract Sifter users that over the years have given me feedback and helped make it a better tool. So what I'm not going to talk about today, but Christy just told you about, so that's great, is I'm not going to talk about the EPA chemicals dashboard too much, but in an annual meeting for ASCCT, there is a whole day where my colleagues from the EPA and I will be talking about the chemicals dashboard. And I'll specifically be talking about the Abstract Sifter and the parts of the dashboard that are synergistic with the Excel Abstract Sifter, and is actually an implementation of the Abstract Sifter on the dashboard, and it has some differences which I'll go over then. So sign up for that meeting if you're interested. So a little background on the field that I work in, literature informatics, and it's a field that really helps people make any literature task easier and more friction, get you things faster, easier, better. And I certainly recognize when I say that there are lots of literature tasks. And all of this from one day to the other may have different goals. When we approach a biomedical literature, we may have a quick question we need to answer. We may have a long, complex literature review we need to write. All those things fall into the purview of literature informatics. And you may be able to see today how the PubMed Abstract Sifter can help with any of those tasks. The most important one, though, is document retrieval. And that's putting a query somewhere and getting some kind of a document back, whether it's a website or in this case, in PubMed, an abstract or a citation. And I'm going to use the word abstract, citation, and article probably synonymously through this presentation. The Abstract Sifter does its document retrieval from PubMed, which is, as most of you know, a great resource funded by the US government, National Institutes of Health, and the National Library of Medicine. It's been around since the Civil War, and it has now over 30 million citations. And just to give you a flavor, and because I love trivia, I'm going to pose a trivia question, which you can all answer in the privacy of your own home and your own head, because obviously we're remote here. So here's the trivia question. What year is the earliest citation about toxicology in PubMed. I'll give you a couple seconds to think about that, and then I'll tell you the answer. It's 1781. Yes, the London Medical Journal, and here is an article about a case of a boy poisoned by the root of a hemlock dropwort. And if you'll indulge me for 1 second, I'm going to read the first part of this article because it's so fascinating, particularly the syntax. June 9th, 1781, the eldest son of the Reverend Mr. Kirkpatrick, a dissenting minister about 9 years old, rambling with several other children, ... If you want to know the rest of that fascinating story, you can look it up on the Abstract Sifter. Okay, back to the serious stuff. PubMed is a great resource, but it has some limitations and frustrations. And the original aim of the Abstract Sifter was to just basically find what you want more quickly and without the frustration of dealing with the PubMed website. And then over the years, more functionality has been added. The basic concept behind the Abstract Sifter is that you're working in Excel and you compose and send a query. It goes via the interweb up to PubMed using APIs or web services. PubMed gets your query, figures out how many citations satisfy the query, and then sends them back, parses them, and places them in the spreadsheet. So you may ask, well, why Excel? In the EPA, it's pretty much a standard data exchange tool for us. We use it all the time for data. And you would think, well, PubMed articles aren't data, but in the Abstract Sifter, we're kind of turning PubMed results into data. Excel is familiar to most people. Most people know or many of the different features that it has built into it. And behind the scenes, there's this VBA programming language that you can use to add functionality. And last but not least, Excel gives you a file, and that's great because with the Abstract Sifter, you can rename it. You can have an Abstract Sifter for every project you're working on. You can mail it to somebody, you can put it on a shared area, and you can attach it to a publication as supplementary material. All those things are possible because you're working with a file. Now, the downside is Sifter will not work on a Mac. So I'm sorry, but that's not my decision. That Apple doesn't quite support the language, the VBA programming language behind the scenes the same way that Windows does. So my apologies for that. So let's dive in and see how it works. I'm going to open up version 5.5 right here. And I want to tell you that when you open up these, because they are macro-enabled Excel files, sometimes you'll get that bar up here, sometimes you don't. And then you have to click on a little button that says Enable Content, Enable Macros. So now I have the Abstract Sifter open. It is an Excel macro-enabled file, and I'm going to go show you real quickly kind of the high-level structure. You come down here, you see all the different sheets that are in the file. Here's the README sheet. Here's where I kind of keep notes about the changes I make to different versions. And at the top of the sheet are links to YouTube videos, instructional videos, the basics of how to use it. And here's that FTP file link. The main sheet is the one I came into. This is where you'll probably be spending a lot of time if you use the Abstract Sifter, because this is where you are talking to PubMed. The abstract sheet I'll get to. The log sheet is another important sheet. Every time you run a query on the Abstract Sifter, a record gets inserted into the log sheet saying what the query was you ran, how many records you got back, and when you ran it. So we'll get to all these sheets again, and I'm going to hold off on these for right now. Go back to the main sheet and jump right in and click this button up here that says Query PubMed, and I'm going to put in just a very simple query. My examples today are going to be mostly chemicals examples, but PubMed has tons of other information. It has nothing to do with chemicals. And so you can put any kind of query you want to in. I'm going to pick a very familiar chemical. Fipronil is the tick and flea medicine we put on our animals, and it has a nice corpus of information about it. So when I clicked on submit, this query was sent up to PubMed, and I was too slow to show you, but down here there's a little, can you see this last line here which says complete? This is the sort of status bar for Excel, and it'll tell you where you are in the query. So we brought back 1352 records from PubMed, and they were all inserted here on the main sheet. The columns that we have are PubMed ID, hyperlinked. These are the Sifter terms. I'm going to jump over those because I'll come back to them and spend a lot of time with those. We've got the pub year for the article, publication year, the title. These two columns are flags. This one is one if the article is a review. This is one if it's in PubMed Central. Okay. Can you all see this? Do I need to move this thing out of the way here? Down here. Okay.
No, we
Here
can't.
The authors, the journal.
Panel, so we can just see your slides. Yeah.
You can't see the control panel thing.
No.
Okay. Just hyperlink the DOI, volume issue pages, and the publication type. So just the basic citation information for the article. And I'm going to do Control N, which is Excel's way of getting to the bottom, and you can see we have our 1352 citations right there. And that's great. That's wonderful to have them here on the main sheet, but still there's a lot there and it's hard to find what you want to find. I'm going to show you now how to use the Sifter terms. And these Sifter terms are things that you type in, and you type into one of these columns. I typed in Sifter term right there, but just as a placeholder. So on B3, I'm going to type in a search term, a Sifter term. And when I'm looking for chemical information, oftentimes I will actually put the chemical name right here. Okay, when I typed in fipronil into B3 and then moved and hit return, what happened is the abstract sifter looked at this string of characters, FIPRNIL, and counted the occurrences of that string of characters in the title, abstract, and keywords of every citation that I brought back. So this is a measure of relevancy. If I really want an article about fipronil, maybe I don't want one that only has one occurrence of the term fipronil. Maybe I want something that has, say, 26 occurrences of fipronil. This is a really fast way of pulling stuff that is relevant to you in that moment. Okay, so there are 26 occurrences of fipronil in this title and abstract and keywords. Let's see, let me go over here to the second Sifter term, and I'm going to just click on that and type in, maybe I want rat studies. I'm going to put rats. And again, as soon as I hit return, the Sifter goes out and counts all the occurrences of that string of characters and all the titles, abstracts, and keywords. I can just click on this and show this is the actual formula that gets, and then I'm going to come over here, and maybe I'm interested in rat articles about fipronil in rat livers. If I sort on these fields and do this filtering thing, I can find a corpus of articles that is about fipronil in rats and livers. I can change this. The Sifter terms, you can use them over and over and over again and put in, say I want to switch to kidney, put that in there. Or let's go to thyroid. All right, so this is a great way to just kind of get an overview of the literature and to find things really quickly. Let's take a look at how you can read what's going on. So here's the article about fipronil in rats and thyroid. It has a lot of it really is about fipronil because it has 26 occurrences. So I'm going to double click somewhere in this row. And when I double click on a row on the main sheet, it brings me to the abstract sheet. And you can see that it has colored the fipronil in blue, rats is in red, and thyroid's in green. So I have a way to quickly skim this abstract and find the terms that I was interested in. So again, on the main sheet, find an article of interest, double click, and then you can read it here on the abstract sheet. If I say, well, this is a great article, I really want to remember this article, I don't want to have to find it again, you can add a note here in the abstract sheet. And the notes are really whatever you want them to be, but you can put a colorized flag on it to say, yes, this is really important, and you could just do that. Or you can add a note and put a tag in, which is whatever you want it to be. This is rat thyroid. When I put a note in like that, what happens is on the note sheet, a record gets inserted for everything I take a note on. So that, and if you save your abstract sifter, your work is then saved, and you can come back later or whatever you leave, come back and your notes are still there. Alternatively, you can, on the main sheet, you can select a bunch of rows. Say I like those, and I'll come down here and select those, and maybe this one. Whoops, I don't want to do that. Select those. Select this one. Maybe go down here and select this one. Select a bunch of records that you know you're interested in, and then say take group notes, and then put in just one time. You just put in a tag and a note, and it gets assigned to all of those. And you can click on either of these colors. If I click on maybe, changes the colorization. So when I put a note in, you can see too here that the PubMed ID has the color that's associated with my note. I go to the note sheet, I can see what I've just put in. If you've got work here from beforehand, you can always delete things from the note sheet like that. If you do delete from the note sheet, you're going to want to reflect those changes on the main sheet. And so you click on highlight, noted PubMed IDs, and then your colorization is changed here. The other thing this is something I actually just learned recently, is that with Excel, you can sort by colors. So if you've done a lot of note taking, you can bring up your different colors to the top so that you can see what you've taken notes on. Okay, so let's go back to sifting, which is what I do a lot, because oftentimes you want to slice and dice the literature in different ways. One really important thing to do when you're sifting is if you're looking for dose studies, you could put dose in there. And then you get a good idea of dose studies, because oftentimes with dose studies, it's telling you all the different doses, and it'll say dose over and over again. You can also put in mg per kg, which is a very common way of identifying or delineating what doses you've got. And you can find articles that way quickly. But even more effective, just put in mg, and you get things that say, like here, we have sort of mg per kg and a slightly different pattern. So it picks up those two. The other thing I would recommend for this sort of thing, you can also do things like, look up little terms like that are hard to search for, but are great to sift on. Parts per million is another thing that's very commonly used for, say, water studies, for finding fipronil in water. Oops, let's take that off. Well, maybe I do want to. So using a combination of different sifter terms and sorting, you really have the whole corpus at your fingertips, and you can find things very, very quickly. So that is sifting, and I'm going to go over to the log sheet next. And maybe you've been working on fipronil for a while, and you think, okay, I'm actually, let's go look at some articles about catfish and river pollution. And you okay, I might have done that too fast. Let me go back to the log sheet. You can rerun any query that you've already have in your log sheet just by double-clicking on it. And here's the 2 queries that I ran this morning. And if you want to rerun them, you just double-click on them. You don't have to type them all in. So here are my 95 articles about catfish and pollution. And I can sift and take notes and do whatever. And that was obviously a wrong note. Okay, so I put a note in that wasn't actually right. This is about catfish and water, so I can go over here and delete that and say catfish is actually what that article is about. And now if I want to go back and continue my work on fipronil, I just rerun this. It just takes a few seconds. Again, you can go down here to the status bar to see how far Sifter's got on your query. There we go. And I want my highlighting back because my notes are still there. And I can highlight there, and now I've got my notes back, my highlighted notes back. The other thing I wanted to show you from the abstract sheet, let's look at this abstract, is that there's a functionality called like this up here. And if you click on that, it will either, it'll look for 133 articles in this case, in PubMed, most like this one, or so you can click on get 6 articles that cite this one in PubMed. And you can append it to what you already have out there or delete and then add. So I'm going to go ahead and get those 6 articles. And they're down at the bottom here. Those are the 6 that were just added here. Okay, I'm going to go now and pretend that I've got some great work done on fipronil, and I'm going to go to the note sheet and show you some additional functionality on the note sheet. The note sheet is where you are engaging the most with the literature. It's where you can triage, read carefully, take careful notes, whatever you want to do, or don't, whatever. You don't have to take any notes. You can delete them all, or you can change the tags. You can, it's up to you. This is where your work, the way you work it takes over. One of the other capabilities, though, on the note sheet is if you double-click anywhere in one of the rows, you get this curation form, is what I call it. And here you can kind of take your time, read the abstract, and as we all know, oftentimes abstracts will have a lot of background information, and maybe they have 1 or 2 sentences that are really the point of the publication. And so you can then find that sentence or maybe some data, and here maybe this rat data, and I've decided this is really what the point I want from this publication, from the abstract anyway. I can highlight that and just drag it over to my notes. And now I've got that separated out so I can pay attention to that later on, because I've done the curation there. It doesn't take it out of the abstract. In fact, you can refresh the abstract and it gets back. And maybe this is another important point, and you can click and drag that over there. Say OK, and then you've got that on your note sheet, and you can always go back and read it. Another new functionality is you can then copy that. I'm going to open up Word real quick. So I did a copy, and now I'm going to do a Control Paste. And that record here has been pasted into Word, and here are my notes, the things I thought was important, and I can then go and maybe bold those or something like that, and work this into my manuscript or whatever I'm doing. Another feature that I've just added to the Abstract Sifter is the ability to attach a local PDF. This does not go and retrieve your PDF, but it will attach a PDF that you've already got. I'm just going to pick out one here, and I can open it from here. So let's say here's my, I'm just pretending, but this is the PDF I'm interested in, and I think this is really important text. I can copy that text and paste it here. And then when I go back in, there's the text I took out of the PDF. Maybe I should actually write from the PDF here. Page, whatever it was. So this gives me an opportunity to really curate what I'm pulling out of that abstract. And then my take home might be use this paper in the analysis. The notes row has the PubMed ID hyperlink. It has these flags, which you can change here too if you want. I mean, it's a little bit easier to do it in the curation form. And then it has when you took the note. Here, this is the PDF link that I just put in, and I can click on the PDF link there, and it also brings up the PDF. Remember, though, if you do save this Excel spreadsheet, and then you want to share it with somebody, and you mail it to some PDF link that you are. So that's something that's probably going to be only something that you can use. So that is the note sheet. And again, you can clean it out. You probably are going to have notes sticking around. If you download a new Abstract Sifter, you can delete anything after row 2 here. All right, so that is, oh, I haven't shown you this yet. All right, so let's say you have a bunch of notes and you want to export them. You can highlight the notes you're interested in and say export, and you can export to PubMed, or you can, this is a query that you can put in PubMed if you want to go and look at those articles in PubMed. You can get them comment delimited or by lines, and these are different software from PubMed ID's input. If you click on RIS format, it will download your citations in a format that can go into EndNote. So that's really useful if you're working on a publication and you want to have the record in EndNote. And then you can also export everything to a text file that then you can pull into Word, and you have all your notes and your tags and everything you can have in Word. Okay, let me check the time here. Okay, I'm about halfway done with the hour. So I'm going to pause right here and just kind of review what I've talked about so far. So I've talked about the README sheet, where you just, there's basic information and links to YouTube videos. The main sheet is where you're going to receive the citations from PubMed. Abstract sheet is where you can look at an abstract one at a time, and then add notes or retrieve articles like that. The log sheet is where all the different queries that you've run are saved, so you can rerun them easily by just double-clicking on the row. And the note sheet is where your work, your ideas, your thoughts are gathered. Okay, now I'm going to move to sort of the second half of what can happen in the Abstract Sifter. And I'm going to jump over to a sheet called the landscape sheet. Christy, are you still there?
We are still here. Lots of great questions.
Okay, I just want to make sure that I'm not talking to dead air or something like that.
Everyone is
weird. Okay, great. All right, so now I'm on the landscape sheet. Why does this keep popping up? And the landscape sheet, I've left a bunch of work out here already because I want to show you sort of the geography of the landscape sheet. And then I will go ahead and build one from scratch. But I wanted to show you what it looks like if you say working on a set of chemicals for a little while. So the landscape sheet lets you get an overview of a set of chemicals. It can be chemicals, it could be genes, it could be diseases, it could be anything. And get an overview of the literature, and then an entry point to start looking at that literature. So we're going to start here with this section. These are my entities in this place. They're chemical names with Cas/RN numbers. And I'll have to confess, I did download these from our dashboard, but they don't have to be downloaded from a dashboard. They could be typed in. In fact, I'm just going to go ahead and type in zip for now. Right there, you can type in all these. You can copy and paste them from something else. Just make sure you don't get any weird special characters. And the other important part of the landscape sheet is this row 3. These are subject matter queries. And these are some that I put in earlier, but I can put a very simple one in like zebrafish. Let's put in zebrafish. This is just a header here. All right, so this area is article counts. So these numbers are the number of articles that satisfy the query built when you take the chemical part of the entity part and append it, here's the lead part, and append it to whatever is up here in row 3. So if I can get that number several ways. Let's go ahead and clear it out. I can, well, here, I'll just do one. Put your cursor there and select a cell, and then click on Update Article Count. When I do that, the query is built, the query is sent to PubMed, and only the count is brought back and placed into this cell. And I can do this for a whole bunch of cells, and then the queries are built iteratively and sent up to PubMed, and the results are brought back. It's a little slow because PubMed doesn't like you to do too many queries at one time, so I have to actually put a delay mechanism in to slow it down so I don't get slapped down by PubMed. All right, so arsenic and zebrafish are in 80 articles. That's the query. If you want to actually see the results, you just say submit and run, and it brings you to the main sheet where you can then start reading about arsenic and zebrafish. Nope, not touch from you. I don't know what it is. Okay, so you can see that's a great way to get an overview of a whole many corpora of literature for different chemicals. And once you bring back your results, you can do things like either colorize it by row, where the highest number in the row is red, or by column, in which case you can see which chemical has the most articles with a certain category of, like I can, there we go. All right, now I'm going to go ahead and just delete all this and show you how to build a landscape sheet from scratch. Okay, so I'm going to do the lazy way right now and just type in for now, and oops. I don't have to have Cas/RN. I'm just doing simple stuff here. And over here, let's do zebrafish again. And start with that. Oops. No. Okay, so those are very, very simple queries. And of course, then I can just select these cells that are in the intersection area between the chemical part of the query and the subject area part of the query, and say update article count. So for fipronil, there are 22 articles about zebrafish. Chloroquine is 29, and atrazine has 57. So that's the sort of manual way that you can build a landscape sheet and do your examination of whatever. There are ways to make it faster. I'm going to show you those next. So you click on the sample queries sheet. Here is where I have put together some very simple queries over the years. There's nothing special about them. They're just very simple queries that you should use as starting points to build your own queries. And I'm going to go ahead and select some of these and then send it to the landscape sheet. All that did was copy it from the sample queries and paste it over here. So all I'm doing is saving myself some typing and typos. If your organization works on queries and tries to get solid queries for a certain subject area, you can put them in there and save them and share them with your colleagues. I've also got some down here that have to do with toxicity. Again, these are just starting points. And so I really encourage you to go ahead and use some of these queries, make them better, get yourself a set that works for you or your organization. And if you you can add them here to the bottom, and just follow the pattern here. So this is going to be the heading, this is going to be the query. Just make sure you follow the pattern here. And then of course you can go back and change. Add things in, food or diet or beverage nutrition, or thinking, I don't know. Obviously, I didn't think ahead when I did that, and then it changes it. Okay, so that's a way to quickly help out with building these queries easily and without typing too much. I'm also going to show you here how you can add to the chemical space. So this is actually a really new addition to the Abstract Sifter, this curated list. And here, these are all lists I've downloaded from our chemicals dashboard. And then sometimes I'll put notes about why I downloaded. For instance, these chemicals here all hit one of our ToxCast assays. These are some chemicals that I pulled out of the Key Characteristics of Carcinogen publications. And some of these, the ones that are downloaded from the dashboard, will have CASRNs, some of them don't. So what you can do here is same kind of similar thing, select some chemicals, and they get then transferred to the chemical space here. And then you can, of course, delete, clean off, whatever you want to do. All right, now I'm going to go ahead and actually delete all these and show you another sheet called the Pathway Query Sheet. This, again, is just a bunch of queries. And in this case, these queries are built. These are ones that I built. And again, it's starting points. There's nothing perfect about them. But I looked at the AOP wiki and pulled off some AOPs and tried to figure out a way to break the AOP into the key events, the molecular initiating events, and the key events. Let me look at this one here. And then for each event, I built a PubMed query that I thought would bring back the literature for that event. So, for instance, this is an AOP for skin sensitization, and the molecular initiating event is covalent binding of proteins. And I picked out some of the key terms that were in the description of the molecular initiating event. So and again, you've got to think, if I'm on PubMed and I want to find literature that describes this key event, what are the terms I might use? Again, these are all starting points. There's nothing perfect about them. So if you're going to use them with a grain of salt and put in your skeptical viewpoint. So if you're interested in one of these AOPs, you can select the row. I'm going to look at this one and send it to a landscape sheet. And then you can go ahead and update the article counts and get an idea of how, in this case, I'm looking at how fipronil might fit in with this skin sensitization AOP. Now, it doesn't have a lot of evidence linking it to a skin sensitization problem. Let's see if maybe some of these other chemicals have more connection to skin sensitization. So now I'm looking to see which ones cause sort of a dermatitis. And chloroquine has the highest number of articles linking it to allergic contact dermatitis. Let's take a look at that. Here's the query. I'm going to submit and have it take me to the main sheet. And here I can then, look and see how it is that, no, okay, I don't need to read now. I just thought, oh, that's interesting. Okay, so then I can read about how chloroquine might cause allergic contact dermatitis. Jump back to the landscape sheet, and I think, okay, well, maybe it does cause allergic contact dermatitis. Is there any evidence linking it to the molecular initiating event? So that query gets run, and there's a lot there. And let's see if there's any literature with the key events. So there might be some evidence there, and that you could dive in and start reading about that. I'm going to delete those and then go back to the pathway queries and look at the key characteristics of carcinogens. Let me just take some of them here and send those to landscape. And these were my carcinogens here. So now the sifter is building these queries one after another, sending them to PubMed, getting the counts back, and inserting the count. Benzidine, let's see what the connection is with benzidine and DNA repair. Again, I'm just looking for some keywords here. Okay. So some interesting stuff there. If you want to do a little more complex thing, you can start out, so I've done, oops, that's not what I want to do. So I got that. Now I'm going to go see, add the DNA literature in. I'm going to append the DNA literature. And append the cell immortalization in literature. See, I'm clicking append here. So I'm just building and building and building all these different steps of the key characteristics of carcinogens. Wait a minute, I'm doing the right. So I did all of those queries and appended the results. I won't do all of them, but there are probably a number of duplicated citations. So another thing that's available here is to just get rid of the dupes. There are 142 duplicated references there, so that cleans it up for me. So I get a nice set of articles that I can start digging into to see if trichloroethylene causes cancer via these key characteristics, or has association with them. Okay, well, I am nearly at the end of the hour. I think we've got, what, 9 minutes left. And I think that I have covered, let me just go through everything in. So the main sheet is where your results from PubMed come back and you can sift to find what you want quickly. Abstract sheet is one at a time. You can read. A log sheet keeps track of everything you did, and you can rerun anything by just double-clicking on it. Notes is where you keep your work and your opinions and your curation results. The landscape sheet is where you get an overview of the literature, and you can use it as an entry point into retrieving citations to the main sheet. And curated list is where you can just accumulate whatever lists of chemicals you want and then send them easily to the landscape sheet. A lot of these are from a project I'm working on now, which is about reference chemicals for retinoid testing, in vitro testing, and I have a couple hundred potential reference chemicals, and I've got them all organized on my curated list sheet. And if I want to examine the literature for one or a set of them, I can just send them over to the landscape sheet from here. I could probably real quick before we end, well, maybe we don't have enough time for that, but there is a way that, again, to get these lists downloaded from EPA chemicals dashboard and to get this hyperlink to the dashboard from Sifter. So I'm going to go ahead and stop there, and if I can answer questions, and then in a few minutes I can just sit here and walk through with you all. If you want to stick around and open up your Abstract Sifter and play around, I will stick around for questions. Back to you, Kristen.
Great. Well, thank you very much. There's a lot of people who are very excited about this tool. Actually, we have a lot of questions, but also just a lot of compliments for the tool that you have built. So kudos to you.
Great.
Let me see here. Just a couple of quick questions, kind of. One of them, a few people have asked, in the Sifter column, like in the main area, can you make more Sifter columns? So.
Okay, so no, in the Excel version, you cannot. In the dashboard version, you can go crazy. Excel's a little more static. You have to stick with 3, but you can change them over and over and over again. So yeah, no. So the answer is no, but try out the dashboard version where you can go wild.
And a couple of questions around, like, how you would, I guess, project management. So if you have something going on for one project, would you save that as that project and then create, open up the blank Sifter again to make a new project? Is that what you'd recommend?
Yes, and I probably have 20 abstract sifters on my computer in each and different project folders. And to start a new project, all you really have to do is you can get rid of the log, delete from the log sheet, delete from the landscape sheet. you don't have to delete from the main sheet because it gets, you know, repopulated every time you run a query. And you can change, you're going to be changing these over and over again. So yeah, you can have as many copies of the abstract sifter as you want to, for one for each project. And that's certainly what I do.
Okay. Someone noticed that, I noticed this too, at one point in the landscape mode, you had Z-fish instead of zebrafish, but I think that was a label. I think it was down where you're pointing,
so
but the actual query was
just label, right? And
yeah.
you can put anything you want to there, but this is where you have to, this is the query section here. So if you double click here, that's what's going to come up, is what's in row 3.
Great. Can you search and sort by author?
Yeah. In fact, I had this is one I've been playing with, and so we all know who Fauci is, and yeah, so you should be able to do any query that you could do at PubMed, you should be able to do here. Now, I have to say that PubMed has changed some of its search algorithms, so, but still, so there you go, some articles by Fauci on COVID. So yes, you should be able to search. Oh yeah, and here's the 1781 year date of publication that I was talking about in the trivia question. So here are all the citations that come up if you search on the year of 1781. And where's the hemlock article? Here it is, right here. Oh, okay, hemlock right here. There's no abstract, but this is the article I was talking about in my trivia question. and there's the first. Yes. So I don't know what question. Yeah. Okay. Yeah. Right. So so you should be able to do any query. That was one based on the year.
Yep. There's quite a few questions about you know, partial terms, like for example, if you put carcino, will it pick up carcinogenicity, or are there sort of Boolean searches that you can do, partial names of chemicals, that kind of thing.
Well, so let's look at, here's one that has the asterisk. Now, I have to say that to use PubMed effectively, you need to learn how to use PubMed effectively. So we're kind of this middleman passing PubMed queries. So whatever rules apply for PubMed should apply here as well. The one exception I would say is if you send through the abstract sifter some weird characters, like if you're copying and pasting and you've got some weird characters, the PubMed website knows how to kick those out. The abstract sifter doesn't, and the web services don't. But you still need to learn how to use PubMed effectively. The abstract sifter does not help you if you don't know how to use PubMed effectively. So PubMed has a great help site, and you certainly should be able to use these asterisk characters. Let's see if there's anything here. I don't know if we're just not picking something up here or not, but okay. And the other thing you can do, let's check this. Let me go to copy. I'm doing Control C, and this is the other thing that's really important to do. Is you should be able to copy and paste. Okay, so no results are found. Let me just take the chemical off there. Yeah, okay. You should be able to copy and paste into PubMed and get the same results.
Mm-hmm.
So that's a really important thing to do, is make sure your query works in PubMed before you try. If you're doing something complex, try it in PubMed first and make sure it works there. again, the more you know about PubMed, the better off you're going to be. So.
A couple of questions about whether there are limits on the number of hits that could be returned.
Yes, 10,000 is the limit. And it's and to get around that limit, what you can do is, if you have a really big query, you can run the query and then do it like whatever your topic is. I don't know, particulate matter and year publication before 2000, and then the next one, do an append and do it after 2000 or something like that. You really have to have something bigger than 10,000.
Someone's wondering about the version. They're saying version 3 is available from the US EPA website, but I don't think the FTP site link that we sent out has version 5, the same version that.
Well, I have to admit, I just changed that recently. So let's go check. So since we're talking about it, let me just, can you see the dashboard here?
Yep.
Okay, so you should be able to go to download and click on PubMed Abstract Sifter, and then this brings you to Figshare. And here's the zip file. No, what am I doing? Register. No, I don't want to do that. I did this yesterday. Just download it. Just download it. Oh, use the value version. Okay. Okay, so yesterday, I think it was yesterday, I did you can see I've been downloading a lot. I did download it yesterday, or upload the new version yesterday. So I've got too many things downloaded here, but it should be the new version that you get from the dashboard now. If not, send me an email. But actually, I will go check that. But I did upload the new version yesterday.
So while you're here, someone is actually asking to show us how to use it via the dashboard. Is it?
Okay, let's do that. Okay, you put in, with the dashboard, you got to start out with a chemical. Someday that will change. So you'll be able to go straight into the abstract sifter. But right now, you've got to go in through a chemical. And it's a little, yesterday, it was a little slow. Okay, so here I am in the fipronil page. And you scroll down to literature. And then click on PubMed Abstract Sifter. And scroll back up again, if you've got it. All right, so this, here's the query so far. This will look familiar, right? And then over here, you can pick a topic area if you want. I'm going to click on water and exposure and say retrieve articles, 145 articles, and here they are. And this is the Sifter terms section, so I can say water, fipronil, soil, dust, kick. I can go on and on like that. And here, the Sifter terms I type in here appear here as column headers, and then you can sort on them here. So it's really nifty. And then if you want to see the abstract, you just click on a row, and the abstract is down here with the colorized terms. Now, that's all you can do on the dashboard. You can't take notes. You can export. You can say select a bunch of things and export it to Excel, or send them to PubMed if you want to then send it to your citation manager. But there's no note taking. There's no landscape sheet. It's just sifting.
Mm-hmm.
So it's great for those really quick things you're doing. is, it's great for quick lookup.
Or if you wanted many.
But if you're doing a long
terms.
If you what? Say that again?
If you wanted many.
If you want lots of sister terms.
Yeah.
Yeah. But for a project where you're going to sit and read a bunch of abstracts and take notes, and then it's much harder. I mean, I would never use it for that. And it's, you can't save your work. Exactly. You can download some things, but you can't, it's just a very different way of working. And it's a much more of a fluid kind of, I need to find something fast kind of thing. And of course, you can add things over here. You can change this in any way you want to, add terms. These, again, are just starting point queries that I happen to put together. Well, but since I'm here, I also want to show one of the other things you can do. You can go to list of chemicals, or you can put your own list in. And let's say we want this ICVAM list of lymph node assay chemicals. Here they are, and I can send them to batch search. And here they are here. I know I'm going really fast. I'm going to click on download chemical data and then click on Abstract Sifter. Yeah, this happened to me yesterday. It's we just rolled out a new version of dashboard, and sometimes you wait a long time. But I can just tell you that what's going to happen is I'm going to get an Excel file when I download, and that Excel file is going to look like this. There's going to be a section of it that's going to look like this. It's going to have the DS Tox ID, hyperlink, preferred name, and the query, and then I can just copy it from that file into either landscape sheet or here, and then it can go to the landscape sheet. But the dashboard is taking a while.
It's working. It's summertime. Couple of questions about the full text. So this is searching abstracts. Is there plans to try to make it cover full text, or is that possible?
Well, I get this question a lot. So full text is a very fraught kind of question, because some of us, depending upon our organization, some of us have access to some full text, but not others. And, it's hit and miss. And so there is a way, I'm working on a way to try to do what EndNote does. If you're familiar with EndNote, you can go to EndNote and say, please get me all the PDFs I have access to. Well, and somehow, I don't know how EndNote does this, and I'm trying to reverse engineer it. EndNote seems to know what your library has access to. And with everybody working at home, it becomes even more complex, because we're not sitting at work on a particular IP address. And unless we're on our VPNs, I'm not sure how we get through the library permission system. Anyway, it's all very complex. Mm-hmm. But the answer basically is you have to download your own PDF at this point, and then you can link to it, but you still have to download it, because what PDFs you have access to it differs for whether you're at home, whether you're at work, what organization you work for, etc. Unless it's a free, PubMed Central PDF or otherwise freely available.
Well, if you have,
I know that's a tough.
Would a workaround be to do your searches in Abstract Sifter and send the results to PubMed, and then PubMed can get your PDFs?
Yeah, EndNote can get, so yes. So the way that would work is you put on the note sheet everything you want a PDF for. Let me just go ahead and. Okay, so everything you want a PDF for, and then highlight them and export them to RIS format, and then that's the next step, and that creates a RIS file for EndNote. And then you can go into EndNote and tell EndNote to get the PDF And then EndNote somehow will talk to your library system. I don't know how it does it. It seems to know what you really have access to as far as PDFs, and then can download them. But I'm trying to figure out how EndNote does all that, but I haven't figured it out yet. If anybody has any idea.
Maybe there's someone on from EndNote. So, you know, we've had quite a few questions. We've been able to answer many of them, but I think maybe we should move to the kind of last portion of the webinar. So, if folks are interested in you know, trying it out and are trying it out on their own machine at home, I'm trying to think of an easier way to let people easily ask questions.
Well, why don't we do this? Why don't I just walk through a couple of very simple things on the main sheet, on each sheet, and then people can try them at their homes, and then send a question at the end of each. You know, maybe I'll do two minutes on, again, you know, how to use the main sheet, give people a chance to try it, and then I don't know if it's going to work, but
we could try raising hands if people if people want to, are trying it out and you want to kind of verbalize your question, go ahead and raise your hand, and we can try that.
Okay, so I'll just go through some really simple stuff, and you guys at home who want to follow along. If not, thanks for coming, or you can just watch if you want to. So the easiest thing to do a simple query is to just go to the log sheet and find something that I've already done a query on, just for fun, and pick one. Let's see what would be interesting here. Actually, let's do something on COVID-19. Something like that. Something that's of topical interest. This is so weird. Okay, so I've only brought back a couple hundred articles, but I was interested in like what kind of medications people are using.
Maybe therapy, would that pull out?
Okay, what?
Therapy, yeah, drug, that would work.
So here's an article about chloroquine, hydroxychloroquine, with or without, people with or without diabetes. Okay, so you at home, your assignment is just to do a simple query, and then to put some terms here in the sifter boxes, and just keep on going. So chloroquine. Did I spell that? Oh, yes, I did spell it wrong.
One person's raised their hand, Surya Hari Lal. I'm going to unmute you, see if you want to ask your question.
Hi, hello.
Hi, Surya. Okay. Well, let's go to the next person. We have Shagun Krishna. Do you have a question?
Yes.
Welcome. Go ahead.
Can you? Okay. I want to ask that what if we want to include the epidemiology studies also? So what terms should we use? Like just because sometimes they are not epidemiology, they are human or.
Well, the same that you would use, if I understood correctly, you'd use the same query you would use in PubMed. As a query, I spelled that right? No, maybe not. Okay, I think that's just so many. And
it's
clinical. I don't know. Yeah. So what I've noticed is if you're doing an epidemiology study for toxicology, if you sift on things like human, subject, worker,
okay.
That's often a really good way to find the studies about humans, particularly if it's a workplace kind of. There's some kind of a toxicant at the workplace. There a worker OCCUP is often something that's good for occupational articles. You know, just a little worker. What else? Farm worker. If you're looking at pesticides, you can look for farmers. This one's taking a little bit longer. Sorry, this is like 3. Okay, now we're 500. I've just entered a pretty big query here, and 1,500, 2,000. Sorry, I such a large query here.
I get it. Probably you can cancel it because it's going to take a lot of time and resources.
It's almost done. It's almost done.
Okay, and next thing I want to ask that how many chemicals can landscape handle at a time? Suppose I have a list of 300 chemicals. I already collected 300 chemicals that are known to be cardiotoxic, and I want to see that what are the articles that are reporting their in vivo, in vitro, or epidemiology. So can I provide a list of say 500 or 300 chemicals, and it's going to do for that, or is there a limitation of number of chemicals?
Well, the limitation in Excel is the number of rows, which is, I'm sure, it's probably 32,000 or something like that. But you're not going to want to run 32,000 queries, send 32,000 PubMed. So what I have done in the past, when I had, say, hundreds of chemicals, I will just, load the chemicals up, save my spreadsheet, and select a whole bunch of cells, and then click on update article counts and come back the next day. Because, it's going to take a couple seconds for each one to come back, at least a second for each one to come back. But the only limitation there is what PubMed's going to let you do. And PubMed does have some limitations on how many calls you can send to it. I've tried to fly under their radar by putting a pause in between calls. But really, the only limitation is PubMed at that point, because Excel can handle thousands.
Okay, I have a last question. Hope you have some time.
Sure.
Okay. Okay. Yes, I'm also working on collecting on a reference chemical for for evaluation of cardiotoxicity, and I work at NTP. So, is there a way that I can simply say that it can simply retrieve those articles which have environmental chemicals, or like I can just enter cardiotoxicity and I can get the chemical name, or
are you talking about downloading the dashboard?
Yeah.
All right, so if there is we're still waiting for the dashboard. Okay, so one of the other things to do, if you have a set of chemicals at NTP, you can go to the batch search, and if you have, I don't know if you have the CAS or any of the names, you can, if you have the names, let's say, you can paste them in here. If you have, say, a list from some document you're working on, whatever it is, just paste it in here. Okay, I don't know why it's not letting me hit enter, but And then you can download chemical data here. So what it will do, it'll take their chemical names, go and find the DSS Tox ID, and create records in a spreadsheet that you can then copy and paste into the abstract sifter. So you can start that way as well if you have your own list that's not on the dashboard.
Okay, thank you.
Yep.
Thank you, Shagun. So a couple of questions I think are, this is a nice question. How do you know what terms to use in a query? And at first glance, it seems kind of obvious, but I think you and I have worked on a project where we actually had a group of people get together and brainstorm terms to use in a query, and really found it to be an iterative process of figuring out what are the right terms that find exactly what we're looking for. Do you want to talk about, maybe some of the projects that you've done and how you figured out what the right query terms are?
Yeah, so I have 2 comments on that. So if you're one of the nice things about the abstract sifter is that when you're doing something like going after a chemical, the information on a chemical, you don't have to worry about being exact about the query. You just go after the chemical name and maybe its synonyms, etc. But when you're doing a query like this, where you're trying to really tease out some biological aspect of what a chemical does, then it is kind of important to have the right terms. And you're right, Christy, it's really iterative. I worked on a project having to do with embryonic stem cells, and we actually went to 5 different experts and said, okay, what are the articles you consider to be absolutely essential to this field? And so we changed our query until we pulled up all those articles. So that's one way to do it. Or you get a bunch of experts and you look at a bunch of articles, sort of hit the nail on the head, and yeah, you talk about it. And it's okay to with the abstract sifter particularly to kind of go broad. You don't have to have precision in the query. You do have to have recall, which is the term for picking up everything you want. But with the sifter terms, you can then hone that precision and find very quickly the information you want. But again, as I'm going to say this again, learning to use PubMed is a really good skill, and it's PubMed has a lot of advice about how to use it. And your librarian, your organization usually has a librarian who's taken a class, probably, in how to do good queries. So take advantage of that. I certainly have, because I have never taken a class in it, and so it's an important thing to learn. But with the abstract sifter, you don't have to spend a lot of time up front. It does help, but you can just dive right into something and learn as you go, and keep making your queries better and better as you go along. And I will say too, particularly on these ones that I pulled from AOPs. So I might take, in these cases, I might go to the AOP and read the key event and take out some keywords from the key event described in AOP Wiki. But then when I start reading about the key event in some paper or something, I realize that the AOP Wiki simplifies this key event enormously. And maybe you've got a molecular event in a key event that really is a cascade of 15 different molecules banging up against each other and phosphorylating God knows what happens. And the more you read, the more you realize that it's not just IL-16 that's in there. It's IL-17, and NF-kappa B plays a role. And you can then start to expand your query the more you read, and the more you find these entities who are actually playing a role in your adverse outcome pathway. Now, it may not be what the actual composer of the AOP said, but if you're just doing literature, it's certainly worth expanding upon these key events.
So sort of similar line of wondering about synonyms. So, what about drugs that have different names in different countries, or, human versus Homo sapiens? How is that handled? Would you have to put in humans and Homo sapiens?
Well, so PubMed, when you do a query at PubMed, they do a lot of expansion. Again, this is something you should go read about. The PubMed articles have mesh terms, and whenever you put a query in a PubMed, it tries to take whatever you use as your query term and expand it. So if you put in fish, it might expand it to all different species of fish. And you can check that. Here, let me go here. Okay, so I did a very, very simple query, fish. Well, it took the fish and made it into fishes. That's a mesh term. And then, well, that's not a very good example. Let's take this one. Okay, here's the one, the endothelial cells. Look and see, it did all this expansion, and it will do things like use the mesh tree to find things that are more, that are in the mesh tree. So probably for fish, it will pick up trout because the mesh tree has trout associated with fish. You can't really see that here, but because it's a mesh term, it will do that expansion, or you can stop it from doing the expansion. So again, this is, when you're doing your search, it's really important to know how PubMed works. When you're doing the sifting, that's really dumb. That just looks for a string of characters. So if you're looking for 19 for COVID-19, you can just look for the 19. Now, there might be lots of other things that have 19 in them. That's a really bad example. I'm trying to think of a good example here, but corona. I don't know. You're looking for that string of characters. So this is very, let's say, dumb. It's just looking for a string of characters. But a query to PubMed does a lot of stuff behind the scenes. And I don't do it, the Sifter doesn't do it, PubMed does it. So it's really important to know how PubMed works, because we are piggybacking on PubMed here. I hope that answered that question. Anything else, Christy?
Oh my gosh, I was on mute. I'm sorry about that. So one last question maybe, which is, what are you thinking as future developments for the tool? Where are you thinking of taking it, or, what do you have in mind there?
Well, I will say that within the EPA, we have another version of this tool that's called the Abstract Sifter Plus. Unfortunately, it cannot go public because it does rely on, it lets you go to things like Web of Science, ProQuest, Science Direct, Springer, USGS, and a number of sources that also have web services. They're all different from PubMed, but they do have, other sources do have web services. So I spend a lot of time on that project, and I'm hoping that maybe in the next year, I could have sort of an Abstract Sifter Plus public that will look just like this, except that you would say, I want to query PubMed, oh, I want to query USGS, oh, I want to query PubAg, which is a free service, and just have it let you query a number of different free and open bibliographic resources. So that would be nice. And then we're also working on the dashboard and designing more of the functionality that the Excel version has into the dashboard version. Those are the big things.
I really like the links between the dashboard and the Sifter. I think that could be really useful for researchers.
Yeah, and so another thing that's coming, speaking of synonyms, is the dashboard does keep synonyms. And one of the things, I'm not sure how to roll this out, is a way to go in for a chemical and pull out all the synonyms and then make a query string out of all the synonyms. That's probably possible today, but the other thing I'm working on, I have worked on, is to go through and say, okay, of all these synonyms, for instance, we did a dye project, and some of those dyes have, how slow this is, some of those dyes have 100 synonyms, and maybe only 3 of them are actually used in PubMed ever. So why send a query with 100 synonyms when only 3 are actually ever used? And so it's a way to take the synonyms that are out there and use only the ones that will give you a hit in PubMed. So here are the synonyms for fipronil. And one of the things you can download is, I'm not sure, I've never really used this, but you can download the synonyms and then put an or between them and put them in the landscape sheet.
Oh, yeah.
So, but I also want to warn people that some of these synonyms have weird stuff in them, and you get strange results from PubMed. So anytime you get a strange result, take your query to PubMed and see what PubMed does, and try to keep things as simple as possible, especially when you get these weird synonym names like this, because they often there's occasional times where they will not work if they have special characters in them.
Okay. Well,
keep it simple.
Thank you so much for this demonstration, for holding on to answer a lot of questions. There's a lot of accolades coming in, a lot of appreciation for the tool that you've made available. So thanks a lot, Dr. Baker, for coming on and sharing
thank you,
this with
Christy.
us.
Thanks to everybody.
Yep, thanks all of you.
I appreciate it. And
joining for your great questions, and hopefully you'll be able to join the next webinar. All right, have a great day, everyone. Bye.
Thank you, everyone. Bye-bye.
Thank you.